How many different ways can you hold a hand? Any 3D object can take on a variety of orientations in space, with its structural characteristics dictating how those objects, whether they just be an object, a hand, or an enzyme, can come together. With this framework in mind, in order to further understand how structure dictates kinetic parameters for enzymes, we attempted a machine learning-powered kinetic reconstruction of human metabolism. Direction on this project was provided by Dan Zielinski. The computational work was completed by Kevin, Hong, and Zafar. Genome reconstructions aim to capture an organism's metabolism on a cellular level, which aid in understanding the underlying mechanism of the biological system. This knowledge is necessary for biologists to optimize strain design, such that cells produce new bioproducts in higher quantities. Reconstructions currently map the relationship between gene, protein, reaction, and flux values, but the elementary kinetic parameters used in Michaelis-Menten kinetics are not currently found in the model. Protein function is dictated by its structure. Proteins have the ability to interact with many cellular components, such as other proteins, drugs, and most importantly, metabolites. For the purpose of this project, we wanted to investigate which structural properties of metabolites and proteins dictate kinetic Km values. 3D structural renderings of protein show the folding pattern for different amino acid sequences, but they do not provide much information about how proteins interact with other cellular components. Dynamic simulations are very computationally expensive and difficult to validate with experimental data. Kinetic Km values have been obtained for most studied enzymes. For a reaction to occur, the metabolite has to find and bind to the active site on the enzyme, where the enzyme facilitates the reaction and then releases the product. Kinetic parameters are obtained according to Michaelis-Menten kinetics, where Km can be interpreted as how tightly a metabolite binds to the enzyme. Since there are more than 50,000 reactions with multiple data points for each reaction that require curation work, coding scripts were written to help match the kinetic data to enzyme reaction and structural properties. The structural properties of the protein and metabolites were ordered into two separate matrices. The metabolite data was aggregated into values per reaction such that each kinetic data point had its corresponding features. Machine learning algorithms can help us extract features that are significant from high-dimensional data sets. Principal component analysis can be used to visualize the sources of highest variance for data sets that have more than 2 dimensions. It allows us to isolate features and then plot them in 2D or 3D space, and correlate them with metadata to obtain biological meaning from the components. We created 3 predictive models using linear regression, p-elastic-net, and random forest, with random forest being the best performing algorithm. Human cellular metabolism is a very complex system, which makes it hard to create a machine learning algorithm that can capture the dynamics of the system while still being simple enough for interpretation. While none of the models had high predictive power, this project brings us one step closer to understanding the underlying mechanism.